So chapter 19 is just an extension of chapter 18. And honestly, I'm not really sure why they split them into two chapters. So a bunch of this is going to look kind of familiar. So we're still talking about the urinary system. So we're still going to be talking about the kidneys. We're still going to be talking about nephrons and tubules. But now we're just going to focus a little bit more on balance. Why is this not clicking through? Okay. So we're going to explain the concept of balance, which is not hard. We're going to talk about body water and how that maintains balance. And we're going to be thinking specifically about how water is influenced by osmolarity and how we see that changing throughout the nephron. So these are the sections of the chapter that we're going through in this set of slides. Don't think we'll get through all of this today. But starting with the concept of balance, all we really mean when we're talking about balance regarding the kidneys is that the kidneys are important for maintaining homeostasis. And specifically, we're trying to maintain a water balance and a balance of solutes and, as we'll see, also a balance of acids and bases in the blood. Okay, but the basic concept of balance is kind of just like the concept of equilibrium. We're saying that the amount you put into your body plus the amount you produce of whatever it is that we're tracking should be the same as the amount that you use, whatever that thing is, and how much you excrete. So if we're thinking about water, we're thinking about the water you drink plus the water that's a byproduct of any of your metabolic processes should be equal to the amount of water that you're using in other metabolic processes plus the amount of water that you're excreting. So how the kidneys work is really going to affect the plasma composition and specifically the amount of water and solute present in the plasma, so in the blood. And that's really what we're trying to keep in balance. So we're going to be partially changing these solutes in order to regulate acid-base balance, which we'll be talking about at the end of this unit. And we're also going to be thinking about how the movement of the water and solutes across different types of cells, including connective tissue, cells in your gastrointestinal tract, and also fluids that you give off, like sweat and the humidity in the air you breathe out, affect the content of the plasma. So we're going to sort of map out all the sources of input to body water and output for body water so that we can kind of track where everything's coming from and where everything's going. So here we see a map. It's from your book. So what we're trying to sort of keep in balance is everything going in and everything coming out. So here in the center, we have the plasma and interstitial fluid, which is really where we're going to be thinking about holding most of our water. So we're going to see that some of what's in the plasma and the interstitial fluid comes from ingestion. So when you eat something or drink something, it goes into your digestive tract. Eventually, stuff gets excreted as feces, but along the way, as it moves through your digestive tract, specifically, mainly through your small intestine, also your large intestine, you're going to be absorbing various solutes, various nutrients, some water. That's going to go into your plasma and interstitial fluid. You're also going to secrete things back into the digestive tract, like bile, and you'll be secreting things like insulin. So stuff is going to come out of the plasma as well in that process. And then your plasma can exchange solutes and water with cells and with extracellular connective tissue. So this will be important as we start to talk about like calcium. So calcium is important for your body, and that's really one of the things that's going to be exchanged with bone. And then here, what's highlighted in purple is what we're talking about now, which is exchange with the renal tubules, so the nephron, so the kidney. So we talked about how we have pretty much everything except for proteins being filtered into our renal tubules. lots of stuff gets reabsorbed back, specifically water and all of the glucose. Then we also have secretions, so like kind of extra stuff that we push into the renal tubules. And then we have two more sources of loss. So you can lose things, mainly fluid, by sweating, hemorrhaging, breathing, because our lungs are moist, so we're breathing out a little bit of water. And you also lose things by excretion and urine. Okay. So when we're talking about balance in physiology, we mean this sort of specific thing about our inputs and our use really equaling our outputs. So here we have water drawn in so that we can kind of understand the concepts of positive balance and negative balance. So when we're in balance, it just means that everything coming into the body or being produced by the body is all either getting used up or excreted. So the amount of total water in your body at any given time stays constant because you add some to it, but at the same time, you're losing stuff at the same rate. So that's balance. We're seeing no change in body water. But sometimes you do have an increase in body water. So you can have swelling. You can be overhydrated. So if we are inputting more water than we're excreting, and if we have solute or water, in this case, in this image, entering the plasma faster than that water is leaving through urination, this means our total body water is going to increase. And when it increases, we say we're in positive balance. So that just means the input is greater than the output. Negative balance is just the opposite. So if we have solute, say, I don't know, sodium or water, again, as in this image, exiting the body faster than you're taking it in or producing it, that means you're in negative balance and the total amount in your body is decreasing. So negative balance is just your output is greater than the input. Okay. As we're thinking about the balance of solutes and water, we're going to be kind of talking about two different types of cells called principal cells and intercalated cells. The intercalated cells are really going to come into play when we start to talk about acid-base balance. So what we're talking about now is mainly regarding the principal cells. So the principal cells are cells that are going to be exchanging water and electrolytes in the distal tubules and the collecting ducts. So these are cells that are sort of specialized for regulating those two things. We should hopefully remember that those two things are kind of what's going to control osmolarity. Okay. So we're going to be thinking about how water balance and osmolarity is controlled. And this is also going to involve antidiuretic hormone, or ADH, which we mentioned once way back when we were talking about the endocrine system. Okay. So osmolarity is tied to the movement of water and the movement of solutes, because we remember that when something has a high osmolarity, it really means it's pulling water towards it because of a high concentration of like impermeable solutes. So water reabsorption in the proximal tubule, so that first part of the nephron, so after we exit the renal capsule with the glomerulus and Bowman's capsule, we're in the proximal tubule, we have water being reabsorbed, but we remember that this is kind of an isoosmotic gradient. So we have both water and salt moving out in the proximal tubule. As we go through the rest of this set of slides, we're going to start talking about what happens when we go down into the loop of Henle, which dives from a part of your kidney called the cortex to the medulla. And specifically in the medulla, we have to really focus in on osmolarity, and I think it's kind of complicated, but there's an interesting osmotic gradient in the medulla that controls how water is going to be reabsorbed. And that's really what ADH kind of plays with in order to regulate water balance. So we're going to talk today, hopefully, about how the medullary osmotic gradient is created. And then we'll also talk about how that affects the reabsorption of water. Those two concepts are really tied together. Okay. So this figure is pretty much like that figure when we were talking about balance generally, but it's just specifying numbers for water balance. So usually when you're in water balance, which hopefully you are, your water intake plus the amount of water that you're producing through cellular metabolism is equivalent to the water output and the water used. So your intake through water, partially coming, well, really almost entirely coming through your digestive system. So you're drinking water. There's also water in your food. Average intake of water is about 2.2 liters a day. I think I drink more water than that, but who knows? So that goes into your digestive tract and it gets absorbed into the total body water. So it's being absorbed across the membranes in your intestines into your body water. A little bit might be secreted back into the digestive tract, just as we're secreting like bile. A little water might be involved there. Very little of the water from your food gets excreted in feces. So it's like 0.2 liters per day. Poop is like a little moist, but hopefully not very, or you have diarrhea, right? So our other source of water is going to be from cellular metabolism, which is a small fraction of the water in your body. It's only 0.3 liters per day, but honestly, that's more than I would have thought of your body producing. You don't usually think about your body producing its own water, but it does. Okay, so the total body water that you have is on average 42 liters of water at any given point in the day. So as we add to it, because we're in water balance, we're not going to be raising that 42 liters up to like 45 or whatever. We're going to be keeping that in balance because we're going to be removing water at the same rate. So our output comes in 4 types. Let's look at the kidneys first because that's what we've been talking about. So water is exiting that total body water because it's being filtered out of the plasma in the nephrons, right? It's going through the glomerulus, that capillary bed, getting caught by Bowman's capsule, going into our renal tubules. So that's this filtration part. A little bit might get secreted. A lot of that is going to get reabsorbed into the body because we remember that we have 180 liters of plasma moving through the nephrons. We don't want to lose the body water. So we're going to excrete some of it, but hopefully very little. On average, about 1.5 liters per day gets excreted as urine. So the other types of output we have, sweating's pretty obvious, right? Like we all know what sweating is. I don't have to explain that. But this last one, insensible loss. So that's you losing water, but you don't notice it, right? So that's mainly talking about the water coming out of your body as you breathe. So this is just a zoomed in picture. Since I was pointing to all the things as I said them, I'm not going to run through it again, but you can see here that it's added up for you how much your water input is. So this 2.2 plus the 0.3 gives you 2.5 liters a day as your water inputs. And we can see that if we add together the water outputs, if we're in water balance, which we want to be, it's the same amount as we were putting in. Okay. So when we're in balance, we call that normovolemia, which I think is a funny word, right? So it's just the normal amount of blood volume. Volemia is always really referring to our blood volume. So we can have hypervolemia. So if you have an extra high volume of blood, if you have too much blood as a result of positive water balance, so you're intaking more water than you're excreting, we call it hypervolemia. And that can be a problem for your tissues. It's uncommon, but like water is toxic if you drink way too much of it. People have died. There was a contest when I was in like high school, so you guys were probably babies, but you know, they had a contest that you would win a Wii if you drank the most water and didn't pee, and someone died and another person got put in the hospital because having too much water is a problem, because that water then gets into your cells, causing them to swell, and when you have swelling in your brain, that can be deadly. Okay. Hypovolemia is just dehydration, basically. So when you have a low blood volume, not enough blood, because you have a negative water balance, so you're excreting too much water, or you're sweating too much, or you're hemorrhaging, because both of those would also make you lose a lot of water. So we know that dehydration, like in extreme cases, especially when we're thinking about hemorrhaging, but also when we're thinking about really being in a really hot environment and sweating way too much, that's also going to cause damage to your tissue in like similar but opposite ways. If you have low blood volume, it's again going to change like the size of your cells and going to cause problems. Okay. So this is largely because of the process of osmosis and the way that your blood changes in osmolarity when you take in too much water or you lose too much water. So water diffuses down its own concentration gradient. So where there's a lot of water, it moves to where there's only little water. And that's always going to be the opposite of where we have our concentration gradient of solutes. So we want to remember that if we have like a low concentration of sodium, and obviously it's not just sodium, it's going to be all your impermeable solutes. I'm just using that as an example because we're like always tracking sodium. But if you have like a low concentration of your solutes, that probably means you have a high concentration of water, because that's what they're dissolved in. And the opposite is also true. If you have a high concentration of solutes, that indicates that you have a low concentration of water. So it's moving from an area of high water concentration to low water concentration, but that's the same as an area of low solute concentration to an area of high solute concentration. So to sort of like abbreviate all of that in my head, I just think of water as like it moves to where it would like water down your solutes. Okay. When we're thinking about movement across cell layers, and especially when we're thinking about the nephrons, our water reabsorption is going to follow solute reabsorption. So when we move salts, when we move solutes, because we then change the osmolarity and that salt content in the place that we move our salts to, the water is going to flow and follow those salts, those solutes, because it flows from areas of low solute concentration to areas of high solute concentration. So if you move your solutes, you're making that area lower than it was before, so you're also going to have water exit. And we kind of looked at that before, but we'll look at it again. Okay. So this is showing us a diagram of most of our nephron. So our glomerulus and our Bowman's capsule will be over here. So this is our proximal tubule with our tubular fluid, which I sometimes call filtrate. I'll try to call it tubular fluid from now on because that's what's in all your images and in your book. So your tubular fluid moving through the loop of Henle, through the distal tubule, and into the collecting duct, and then into the renal pelvis, into the ureters, into the bladder, out of the body. Okay, so that's what we're looking at here. The normal osmolarity of body fluids, so like the osmolarity of your plasma, and therefore also the osmolarity of like your interstitial fluids, your other extracellular fluids, because everything's in equilibrium most of the time, is 300 milliosmoles. So that just means that there are 300 milliosmoles of solute for every liter of plasma. So basically for every liter of water in your blood, approximately. So usually in most places in your body, there's no real osmotic force for the water to be moving between fluid compartments. Fluid compartments just refers to that intracellular water, the extracellular water, the water in your plasma, which technically I think counts as extracellular. So usually you don't have water moving between those parts of the body, between those parts of cells and the structures surrounding cells, because they're in balance, because they're in equilibrium. Okay. The kidneys, though, are going to be able to change osmolarity, and really they're going to be doing that to keep you in homeostasis. So they're really going to be trying to get you back down or get you back up to this 300 milliosmoles of solute. And they're going to do that by regulating the movement of water. So rather than dumping a bunch of salts into your plasma somehow, we're going to regulate the osmolarity of our plasma and therefore our blood and the rest of our tissues by controlling how much water gets moved back into the blood instead of being excreted. So water reabsorption is going to be a passive process. We're mostly not going to be actively moving the water. What we're going to be doing is moving solutes to draw water into different places. So the movement of water is passive, based on an osmotic gradient. So the osmotic gradient is that pull, the pull of the solutes pulling water towards it. So what we're actually going to set up is the osmotic gradient in order to control the water flow. Okay. So in chapter 18, we talked a little bit about water reabsorption in the proximal tubules. So we remember that the proximal tubules are really our mass reabsorber. And so 70% of the water that gets filtered out from the plasma is going to get reabsorbed in the proximal tubules. And we're not regulating that at all. It's just happening along with the movement of the ions that are getting reabsorbed as well. So we've looked at that before. We'll look at it again in a second. In our distal tubules, so they cross over each other, so that's a pain in this one. More realistic. Okay, so our proximal tubules aren't regulating water reabsorption, but that's where most of it's happening. And then here's our loop. Here we can see our distal convoluted tubule, so it loops back past here. Here we see our distal tubule and then into the collecting duct. So in the distal tubules and the collecting duct, so like the end of the nephron, most of the remaining water is going to be reabsorbed, but this is going to be regulated. And specifically, it's regulated by ADH. Another word for ADH is vasopressin. Okay. So in order to control the movement of the water, we're controlling the reabsorption of solutes. So mainly the way we're sort of toggling the switch or using the dimmer or however you want to think about this regulation of water reabsorption, the main way we're doing it is by changing the amount of sodium that's getting moved. So other solutes count too, but just like with our neurons, like whenever we've looked at a cell before, we mainly control stuff by controlling the movement of sodium. So sodium is actively transported across the basolateral membrane. So the basolateral membrane is the one facing the blood. And this is going to establish an osmotic gradient for the reabsorption of water. We looked at that before in the proximal tubule. I'm going to look at it again right here. Last time we saw this image, I think we were talking about glucose. So we're just going to focus in on the water part right now. So we remember that on our basolateral membrane, which is the part facing the blood, so facing this capillary, that's why we have the plasma labeled here. So here's our tubular fluid, so that's the inside of the nephron. So on this basolateral membrane, we have our sodium-potassium pump. So we're actively transporting sodium out of the cell. So it ends up in this paratubular fluid, this little space between the nephron and the paratubular capillaries. But once we get a high sodium concentration gradient in the paratubular fluid, it just diffuses into the capillary. Okay, so we're actively pumping sodium out of our renal tubules. Because we're actively pumping sodium out of our renal tubules, I want to just note here, that means we have a low concentration of sodium in the epithelial cell because we're actively removing it. So that means that sodium can passively move into the epithelial cells. So it moves actively across this basolateral membrane, but passively across this apical membrane between the tubular fluid and the epithelial cell. Okay. So for the reabsorption of water, we're going to kind of run through this in steps. So the first thing that happens is the active reabsorption of solutes. So active in the sense of active transport. So we're using energy, we're using ATP. So we're actively reabsorbing sodium and other solutes. That's why there's an X and a Y. And if we're talking about other solutes, the active part of the reabsorption, like the place we're using ATP, might be at the basolateral membrane, or some of this active reabsorption, like if we're thinking about the glucose, we'd have secondary active here being reabsorbed at the apical membrane. But really we're tracking right now the solutes that are moving into the paratubular fluid. So most of the active stuff that we're concentrating on is happening here at the basolateral membrane. Okay, so this is increasing the solute concentration in our paratubular fluid and also increasing the solute concentration in the plasma, which means that we're getting greater osmolarity. So we're getting a greater like pull on water as we're raising these solute concentrations here. So water is going to move across these cell layers by osmosis because so if you imagine here, we have our high solute concentration in the plasma. It's high here. So that means we're going to pull water from the paratubular fluid into the plasma. So if we've pulled water from the paratubular fluid into the plasma, this means that we're increasing the solute concentration here, which we're already increasing from actively pumping out solutes. So we have a high concentration of solute here as well, which means that we'd have passive movement of water from in our epithelial cell to this paratubular fluid. If we have this movement of the water in this direction, we're also going to need to keep replacing that water from our tubular fluid into the cell to keep it moving all the way through. So this movement of water is all being powered by the active transport of these solutes because the water is all moving to go water them down because they're increasing the osmolarity. So one solute that we haven't talked about yet is urea. So urea is a permeating solute, meaning it's going to go through our membranes. There are going to be transporters that do that. We're going to talk a little more in detail about urea later on. But urea is a solute that's going to be reabsorbed passively all the way across here. But even though it's being reabsorbed passively, right, it's a solute, which means that we have more solutes in the plasma, which means it's also going to exert a pull on this water. Okay, so that's mostly what's happening in the proximal tubules. As we move into the loop of Henle, we're still going to be controlling the solutes in order to move water, but it's going to get a little more complicated. So we're going to talk now about the establishment of something called the medullary osmotic gradient. So here in this image, I want to make sure. Okay, I'm not going to focus on the image right now because it's on the next slide as well, bigger. Okay, but basically Your kidney has two parts, right? It has a cortex, which is like its kind of shell, and it has the medulla, which is the inside part. And as we go into the medulla, so that central part of the kidney, we have a gradient of osmolarity. So near the cortex, near the outside, we're going to have lower osmolarity than we have deeper in the medulla. So as you get towards the renal pelvis, which is like the place everything's leaving the kidney, it's kind of the sort of bend in the kidney beam, that inside bend, there's going to be greater osmolarity. So like potentially a greater pull on water in that location. And this gradient that we're creating across the medulla is really important to how water gets reabsorbed. And most of the rest of our water, right, we reabsorb 70% of it kind of passively in the tubular fluid, but we're going to be reabsorbing most of the rest of it now. Okay, so here on this image, I'm going to kind of show us what we're looking at, and then we're going to go through talking about how this gradient gets set up. But so here's our nephron, so here's our proximal tubule, here's our descending limb of the loop of Henle, here's our ascending limb of the loop of Henle, there's a thin part, and then a thick part, then we're into our distal tubule, then we're into our collecting duct. So our fluid, so whatever we took out of the plasma, is moving this way through this image. We have our cortex here at the top and our medulla here at the bottom. And we can see that the way the nephron works is that as we enter the loop of Henle, we're going to cross from the cortex into the medulla. Okay. So our cortex is at this 300 milliosmoles of osmolarity. So remember that's kind of the balance we're going to be trying to keep, as we saw a couple slides ago. Okay, but as we dive down into the medulla, we can see that there's this gradient. So as we get deeper and deeper into the medulla, we have higher and higher osmolarity, so a stronger and stronger pull on the water. So the water in this descending limb is going to be responding to this osmolarity. So it's going to be pulled out of the descending limb. But when we look at the thick ascending limb, this is really where we're, a large part of where we're setting up this osmolarity. So in our thick ascending limb, it's impermeable to water. So if it were permeable, we might expect water to also be moving out of this part, but it's not. It doesn't allow water to pass through it. And instead, we're again going to have the transport of solutes, specifically sodium, but also potassium and chloride ions when we're talking about our ascending limb. And when we start to talk about the collecting duct, we're also going to factor in urea. Urea is also going to have an influence on this osmotic gradient. Okay, so we call this sort of system in the loop of Henle the countercurrent multiplier. And the reason we call it the countercurrent multiplier is just that as we have fluid moving down our descending limb, right, that's one direction, and then the fluid loops and it's moving back up the other direction. So at any given moment, we have fluid moving in two directions through the medulla, and that's that countercurrent. So that's why it's the countercurrent multiplier. So it's happening as our descending part of the loop of Henle is kind of interacting with factors set up by our ascending limb of the loop of Henle. So this just writes out what I said a second ago. Sorry, that's my dog. The ascending limb is impermeable to water, but it pumps solutes, sodium, chloride, and potassium. The descending limb doesn't really transport those solutes. Instead, it's permeable to water, so what we're mainly going to see is the movement of water. So that's here on our image as well. So we can see that we have fluid moving here through the loop of Henle. We have water moving out into the capillary in our descending limb. And we have solutes actively being pumped out by our ascending limb. And the reason they have these numbered that way is from a functional perspective, this is all driven by that active pumping of solutes, right? So as we actively pump these solutes into the medulla, it raises the osmolarity of the medulla. So it like raises the pull of those solutes on the water, which is why water moves into the medulla. So that's why this is the first step, and then that's the second step. 3 is like the dividing point here. Okay. So the fluid in our proximal tubule, which we just looked at, is at 300 milliosmoles, and the fluid surrounding it, right, the paratubular fluid, so between the capillary and the proximal tubule, is also at 300 milliosmoles. We remember that that's isoosmotic reabsorption happening in the proximal tubule. But that's not what's happening in the loop of Henle. So in the loop of Henle, we are going to see changes in osmolarity as we move stuff. So in the descending limb, the osmolarity is going to increase as we move down. So the osmolarity of the tubular fluid is also increasing. So as we remove water from this tubular fluid, right? If we remove water, that means the solutes left in the tubular fluid are more and more concentrated, which means we have greater and greater osmolarity as we go down here. So that's what's happening in the descending limb when we're focusing on the fluid. In the ascending limb, we actually have that osmolarity in the tubular fluid going back down. So the reason for that is that as this fluid moves forward, so we can see as it's going into this thick part of the ascending limb, what do we have here? The active movement of sodium and chloride, right? So we're actively moving solutes out of the tubular fluid. So even though it doesn't have that much water anymore, as we remove solutes, right, it's suddenly getting less salty again, not because we're watering it down, but because we're removing some of those solutes that were making it highly osmolar. So that osmolarity of the fluid goes back down as we come back up into the cortex. And then here, as we're back up into the cortex, is where we go into the distal tubule and lead into the collecting duct. So in the descending limb, the osmolarity is initially equivalent. Sorry, I'm gonna pause for a second. No, I'm not. You can't yell at my dog. Sorry. Maybe she'll be quiet. Hey, Lucy, be quiet. Okay. So we're gonna look in a second at how this gets set up, but the total result at the end of all of this is going to be that the fluid in our distal tubule, so after we actively remove all those solutes at the end and put them into the medulla in order to reabsorb water in the descending limb, we're going to be left with a fluid that's 100 milliosmoles. And in the cortical interstitial fluid, we have a osmolarity of 300 milliosmoles, as I've said a couple of times, right? Like that's what we saw in the proximal tubule, plus the paratubular fluid, which itself was part of the cortex. So all of the cortex is going to be at 300 milliosmoles. But the medulla is going to have a gradient across the medulla. So it's going to go up in osmolarity as we move away from the cortex and down further into the kidney. Okay. So this is sort of a set of steps starting from nothing. So starting from the entire kidney is at the same osmolarity of 300 milliosmoles. And it's going to show us like how this gradient in the medulla gets set up. So we're going to look at it step by step. But the real state of the kidney is what we're going to see kind of at this end state. So we're building up how we get to this steady state that your kidneys actually function at in this number 7. Okay. So we're setting up like a thought experiment where we start out with a kidney where everything is at 300 milliosmoles. So our cortex is at 300 milliosmoles, our medulla is at 300 milliosmoles, and our tubular fluid is at 300 milliosmoles. So at this point, everything is in equilibrium. Fluid is entering the tubule, starting out at 300 milliosmoles, it's flowing through it. No water is moving because there's no osmotic gradient. So it's exiting with all the water that was in it and with the exact same osmolarity. Okay. So in order to shift this balance, right, to sort of shift up this equilibrium, we have the active transport of sodium, chloride, and potassium out of the ascending limb of the loop of Henle. So here is our active transport out of our tubular fluid into the medulla. So when we actively transport those solutes out of the tubular fluid and into the medulla, what I want you to note before we look at those green arrows is that before everything was labeled at 300, right? We still have 300 up here in the cortex. We still have 300 here in the descending limb. But now the osmolarity of this ascending limb has gone down to 200 milliosmoles because we took solutes out. So we took solutes out so they're less concentrated, so we have a lower osmolarity. We took them out and moved them somewhere though. So this portion of the medulla here in the center between our two limbs of the loop of Henle is now at 400. So it's at 400 because we moved solute from the tubular fluid into the medulla, so it's gone up. Okay. So because we moved those solutes, now we have a medulla that's at 400 milliosmoles. That's now exerting a pull on the water in the descending limb in the tubular fluid, because that tubular fluid is still at 300 milliosmoles. So remember, if we're comparing numbers for osmolarity, we're just going to move towards the higher number. So 300 is less than 400. So water moves towards the 400. That's the direction this pull is going on the water. So we start to have water reabsorption here from the descending limb. Okay. So after that reabsorption happens, this means that we have shifting in water until the fluid, the tubular fluid in that descending limb, reaches equilibrium with the medulla. So we moved our solutes into the medulla to get it to 400. Then we had water move because the tubular fluid was at 300. So it moves until the fluid in the descending limb is also at 400. So now we can see that our tubular fluid and our cortex, our proximal tubule and our distal tubule fluid and the cortex are all at 300, still just like we set out. We had active transport here between our ascending limb and the medulla. So the fluid in the ascending limb has a low osmolarity. The medulla has a relatively high osmolarity. And then we let the descending limb reach equilibrium. So it now also has a high osmolarity. In this next step, the key thing to note here, the reason they've written this word fluid with an arrow on, is that stuff is always moving through the nephron, right? So what's happened here in this image is that we've just filtered out more plasma, and as it's flowing through the nephron, everything that was in the descending limb here is now continuing, and it's moving into the ascending limb because it's basically getting pushed, right, by this new fluid that's getting filtered out of the plasma. So the fluid in the descending limb here used to be in the proximal tubule. So it's at 300 because it was at equilibrium with the cortex. So it's at 300, and now it's moving down into this first part of the medulla. You can see that it's like halfway down into the medulla here in this image. And then we see the fluid that was in the descending limb before. So this fluid that reached equilibrium at 400 is hanging out here in the bend. And then the part that was in the ascending limb here is in the higher part of the ascending limb here. Okay. At this point, we continue to have the active transport of solutes from the ascending limb into the medulla. So this process that we saw in 1 is still happening now, even though we have these gradients set up, right? This is just active transport. We're using ATP. We keep doing it. Okay. Which brings us to our next image. So I'm going to focus on the ascending limb first, because kind of how these numbers shifted, I think deserves kind of focusing on. So in the last image in number 4, we had 400 and 200 in the ascending limb, and then we pumped out solutes. So the 400 went down to 300, the 200 went down to 150. We keep having some movement, so now some of that 200 escaped and is in the cortex in this distal tubule part. Okay. So before, everything was at 400 here in the medulla, but now we've started to move, so to actively transport those solutes into the medulla. So that's one thing that's changing this concentration here in the medulla. But the other thing that's happening is that we're also having the movement of water. So the movement of water is coming here. And so we start out with this 300 milliosmol fluid in the tubules. It encounters the medulla that was before at 400. So it was moving, kind of gradually trying to get equilibrium with that. So that's why we see that go down a little bit. And the same thing kind of happening here. Here we had a difference of 500 and 400, because before we were at 400 and 400, but we pumped more solutes into that area. And so now we're at a higher osmolarity in the medulla here. So we're going to have water movement between this 400 and 500 part as well. The fluid is all continuing to move. So this 400, as it moves into the ascending limb, is going to be going down as solutes are being removed. Okay. So everything here then eventually reaches equilibrium. So we reach our equilibrium 350 up here near the cortex, down to 500 here at the turn of the loop of Henle. And we note that we're reaching equilibrium between the descending limb and the medulla, but the ascending limb, because we have this active transport, is not at equilibrium with the medulla, because we're actively moving solutes, we're actively pumping solutes here. So we have a difference between this 500 milliosmoles in the medulla and the 300 here. And we can see that as we go up, we have less and less because we're actively pumping these solutes. So this gets pushed to here, more solute gets removed. So it goes lower, it gets pushed to here, more solute gets removed, and so on. Okay. And so this eventually, after it keeps going, we have water continuing to flow because we push all these down, and then that process continues. So like this part gets watered down because we're basically putting in tubular fluid that is pretty watery, right? Like it doesn't have that great an osmolarity, so it further waters this down. But our fluid is getting more and more concentrated as it moves down into this bend. So basically less and less water moves out as we go down into the medulla, which means this medulla is getting watered down less and less. And it keeps going. And then in the ascending limb, we're pumping the solutes in order to maintain the medulla as being salty in order to draw the water. We're actively pumping with ATP. And so this keeps going on and on until we reach our steady state, which is the typical state of the nephrons in the kidney. So we have a gradient that can go from 300 in the cortex down to as much as 1400 milliosmoles as we get deep into the medulla. And so this is the state that your nephron is normally in, with this gradient that was really all set up by that active pumping of our solutes originally in the ascending limb. So we're going to stop there. This is just all of that together. And again, this is an image in your textbook, so you can look at it on these slides. You can look at it on your own later. And we'll pick up with talking about how urea affects this on Wednesday.